morning. So I'm really excited to be here and speak to all of you this morning. I want to thank the organizers for putting this together. I know it's not an easy task, and for inviting me to share some insights today. And as Jeff mentioned, I was tasked with giving a clinical update on new agents and trials in less than 30 minutes. So I've been a little bit selective. I'm going to talk about what's kind of coming down the pike in pediatric sarcomas. I have nothing to disclose, but I will talk about several unapproved therapeutic agents. And I thought I'd start kind of just giving the 10,000-foot view of what are sarcomas. So they're a heterogeneous group of cancers that come from the bones and connective tissues of the body. And this can be anything from lymph vessels to nerves to fat to blood vessels, bones, muscles, ligaments, and tendons. And in the adult cancer setting, sarcomas make up about 1% of all of the cancers, which is obviously a very small proportion. But in pediatric cancer, it's about 20%. And these are of a bunch of different subtypes. I've listed several here. The most common that we see are osteosarcoma, rhabdomyosarcoma, and Ewing sarcoma. But there are others that I'm going to sort of touch on today: MPNST, alveolar soft part sarcoma, and then there's a very long list of sort of less than 1% pediatric type sarcomas that are very rare. So how are we doing in sarcoma therapy? Well, overall, we're not doing that bad. So I'm going to show you some statistics for patients with metastatic disease, which is and relapse disease, which are the two areas where I think a lot more work needs to be done. And so this is data showing patients with metastatic rhabdomyosarcoma at diagnosis. And what I'm showing you here, so this is a proportion of patients who survive, and this is over time. And each decade, we've gotten slightly better. Starting from the 70s, we made a big jump up in the 80s, but since the 80s, really, the overall survival is about the same. So there hasn't been a lot of progress in about 40 years. And it's a very similar story for Ewing sarcoma. So again, overall survival, this is metastatic disease again. Sort of incremental jumps up in, new therapies and supportive care in the 70s to the 80s to the 90s, but in the last decade, very similar, kind of sticking right around the 30% range. And so it sort of begs the question, why hasn't there been more progress? What's the deal? And so I actually went back and I looked at FDA approvals for cancer drugs just for the last 18 months, so 2018 to 2019, and these are all cancers. And I made a word cloud showing either the targets or the drugs or the class of drugs. And so you can see there's been a ton of new therapies that have been approved for adult cancers in that pretty short period of time. And then I looked to see what was on the list for FDA approvals for pediatric type sarcomas ever, and that's the list. And I will say that 4 of these drugs are chemo, and one of them is a targeted agent that was just approved relatively recently, and it's not even specifically just for sarcomas. And I think that there are a lot of reasons for this discrepancy, but I think one of the big reasons is that it's actually really hard to find drugs that are effective, and it's sometimes hard to get pediatric cancer access, patients with pediatric cancer access, so that we can do the trials so we can answer some of these questions. So I know that's a really depressing slide, so I'm going to give you a little bit of hope. So I think we're entering an era of lots of new cancer therapies, and so these are the mainstays of our current therapies. So we treat most patients with chemotherapy and then usually some form of local control with either radiation or surgery or both. And there's certainly been some agents kind of coming up that have improved upon these regular, usual modalities. So there are a couple of new chemotherapies that are under investigation for pediatric sarcoma. There's several radiation modalities that are under investigation. But perhaps more exciting, there are kind of whole new classes of agents that are up and coming. And so immunotherapy is one of the most sort of popular ones to talk about. And so I actually just put up a handful of immunotherapies that are being investigated specifically for pediatric sarcomas. So we've got CAR therapies, we've got antibodies, and all of these are kind of under investigation. Another class a broad class of new agents are agents that deliver drugs in a novel way, so drug delivery agents. And I've put a couple of these here, and these also are being actively investigated in pediatric sarcoma. The next group is targeted therapies, and this is a very long list, and there's a very sort of broad definition of targeted therapies. But these include things like PARP inhibitors or drugs that target a specific cancer-causing element of a particular tumor. It includes HDAC inhibitors, which we're going to hear about later in this session. Metabolic inhibitors, which we're going to also hear about, and then a whole list of agents that target specific pathways within the cancer. And so this is an exciting, and this is certainly not a comprehensive list of everything. And then the last sort of buzzword class that we hear are agents that are precision medicine agents. And so these are also targeted therapies, but I just wanted to point out that there is a big study run through the Children's Oncology Group right now looking at alterations in patient tumors and trying to match those alterations with specific drugs. So I think that there's a lot to be hopeful for. I'm going to highlight 3 agents on this list just to give you kind of a flavor of what's going on in each of these areas. So I'll talk about atezolizumab, which is an immunotherapy, HSP90 inhibitor drug conjugates, which is a drug delivery agent, and then TK216, which is a targeted therapy for Ewing sarcoma. So the first story is about PEN-866, which is an HSP90 inhibitor drug conjugate. And basically, this is a drug that attempts to improve upon irinotecan, which is a mainstay either in the upfront or relapse setting for several pediatric sarcomas. Irinotecan is an effective chemotherapy agent, but its use is limited by a couple of things. So it's poorly bioavailable, which means that it has to be converted into an active form before it can have action on the tumor. And it doesn't all get converted into that active form. And then some of it, once it's converted, gets converted back into an inactive form. So it doesn't get to the tissue very well. And then also it comes with a lot of systemic toxicity, as do all of the chemotherapies, including bone marrow suppression and gastrointestinal toxicity. The active metabolite of irinotecan is this agent called SN38, and that's extremely active, but unfortunately it can't be formulated to be delivered to human patients. We can give it to mice, but not to humans. And so what PEN-866 does is it attaches SN38, which I'm showing you right here, to another agent called an HSP90 inhibitor by a linker, and this forms what we term a conjugate. So HSP90 inhibitors are drugs that were initially looked at as anti-cancer agents. They didn't work that well as anti-cancer agents, but what was discovered in the early studies was that they are kept in tumor cells for much longer than they're kept in normal cells. So, if you measure the HSP90 inhibitor in a tumor cell over time, it can stay there for about 5 or 6 days, but in a normal cell, it's cleared out in a matter of hours. And this is just a visual representation of that. So here's your normal cell, the drug gets in, the drug goes out, but in the cancer cell, the drug gets in and it kind of stays around. And so the question that we are trying to answer is, would an HSP90 inhibitor conjugate have the same kinetics, meaning would it do the exact same thing? Would it stick around for longer? And would that result in superior efficacy? Would it work better than the non-conjugated form of the drug in pediatric sarcoma models? And so in this case, we're comparing PEN-866 specifically to irinotecan. Okay, so the first question again, does it last longer in cancer cells? And just to give you a little bit of background, so irinotecan and SN38, they're inhibitors that basically cause double-stranded breaks in the DNA. And when we see a double-stranded break in the DNA, one way we can measure that experimentally is with this protein expression, called gamma H2AX. And so what I'm showing here is an experiment where we took mice, we grew TC32 Ewing sarcoma xenograft tumors in those mice. I'm losing my pointer, sorry. We treated them either with a single dose of irinotecan, a really big dose, the maximum tolerated dose that the mice can get, or a really big dose of PEN-866. And then we looked over serial time points to see how long did the expression of gamma H2AX last. So how long did we see these double-stranded breaks? And I'm showing you just 2 time points here, which I think kind of make the point. So at day 3, we saw kind of an equal amount of H2AX in both of the irinotecan treated and the PEN-866 treated mice. But by day 7, we lost the signal irinotecan, and it was still there for PEN-866, suggesting that this drug is sticking around and actually exerting its action on breaking the DNA longer than the standard chemotherapy would. And so does this translate into efficacy? Does it mean that it works better in models of pediatric sarcoma? And so what we did for this experiment is we injected mice with a variety of different, each mouse with a different sarcoma. So we had Ewing's, we have rhabdo, osteosarcoma. And we allowed these tumors to get quite big. And then we treated either with the maximum tolerated dose of irinotecan for several weeks or the Pen 866 at various doses for the same number of weeks, and we looked to see what happened. And so this is data from a TC32 xenograft, and what I'm showing you here, so this is tumor volume on the y-axis, and this is days post-injection of the tumor on the x-axis. The arrows are showing when we treated, so we gave these treatments once a week. The blue are the mice that are untreated, so you can see they very rapidly progress. The red is just an HSP90 inhibitor alone. It's in there for a control. It's called gantenerumab. Again, no anti-cancer activity. And what you can see is that for the irinotecan and the Pen 866, which used to be called STA 8666, all of the tumors actually shrink down to non-detectable within 20 days of injection. But all the mice that were treated with irinotecan relapse by day 50 or 60, and there's a dose-dependent relapse for the other mice. So the mice treated with 50 mg per kg relapse around 60 to 70 days, and then for the mice treated with the 2 higher doses of the drug, actually a very big proportion of them did not relapse. So it really looked like, at least in this model of Ewing sarcoma, the conjugate was superior. And we did this in multiple models of Ewing's. We saw the same thing. We did it in a rhabdomyosarcoma model, several of them. We saw the same thing. And perhaps most interesting, we did it in an osteosarcoma model, which really was kind of resistant to the irinotecan, so we never saw tumor shrinkage with irinotecan, but we did see tumor shrinkage with the conjugate. And the reason it doesn't go down to 0 here is because the bony matrix is left behind, but when we looked at these tumors, there weren't any cells. So the exciting thing is that now Pen 866 is under evaluation in a clinical trial. So there's a phase 1/2A open-label multi-center study to look at safety, tolerability, PK/PD, and anti-tumor activity in patients with advanced solid malignancies. And this is a first-in-human study. The phase 1 is asking what is the maximum tolerated dose, and also how do the toxicities compare to the toxicities we see with irinotecan. And then the phase 2A is asking what is the response rate. So what is the proportion of patients that have either stable disease or disease that shrinks? And this is going to be done in expansion cohorts. So just to give you an overview of the study design, the phase 1 is a dose escalation, meaning we start at a very low dose because it's a first-in-human study, and we go up until we see that patients cannot tolerate it. And this is in an all-comers adult patient population, so patients 18 and older with any kind of solid tumor. We're looking at safety compared to irinotecan. We're looking at markers of DNA damage in the patient tumors, and then, of course, the response rates. And once we reach MTD, then we'll move into a phase 2 expansion cohorts. And one of the most exciting things about this is that this is a drug that the company is extremely motivated to develop for big indications in adult cancer like colon and lung, but because of the preclinical data that we generated in our lab, there's actually an expansion cohort for Ewing sarcoma and rhabdomyosarcoma. And so we actually enrolled the first evaluable patient early in 2018. We've dosed 17 patients to date, and we're currently still in dose escalation at dose level 7. And overall, the toxicities have been really mild, and we haven't seen the GI toxicity that we see with irinotecan. And we're actively recruiting right now at NCI and would welcome your patients. And I will say we're really pressuring the company to lower this age to 12 once we get into the phase 2A expansion. Okay, so the next kind of mini story I'm going to talk about is one of the targeted therapies called TK216, which targets the oncogenic fusion EWS-FLI1 in Ewing sarcoma. And just to give you a little bit of background, so EWS-FLI1 fusion is basically a fusion between chromosome 22 and chromosome 11, which each carry a gene. So 22 has EWS, and chromosome 11 has FLI1. And there's a aberrant sort of mixing of this genetic material causing a translocation, which is one chromosome stuck to the other. And this creates this fused gene product, EWS-FLI1, which is translated into a protein. And basically, this protein transforms the cell into a cancer cell. That would be Ewing sarcoma. So if it's Ewing sarcoma, it has to have this translocation. And it changes the expression of the genes all across the genome. So it turns on things that shouldn't be on, and it turns off things that shouldn't be off, creating a specific dependency, because normal cells don't have EWS-FLI1. So you can imagine if you can target EWS-FLI1, you'll only be targeting the cancer cells. And many people have looked at ways to do this. So one approach is to say, okay, well, what are the activated genes that EWS-FLI1 turns on and turn them off? Or what are the deactivated genes that EWS turns off and try to turn them on and see if that affects kind of what happens to the cell? Or you could sort of go for the jugular and say, well, we want to just shut down the whole thing altogether, and then hopefully the cell will not be able to survive. So EWS-FLI1 binds to the DNA to do its job of turning genes on and genes off. And one of the sort of proteins that's necessary for this activity is something called an RNA helicase A. So that's showing, I'm showing that in the purple. I don't know if you can see that. I'm showing that in the purple, and this is EWS-FLI1, and this is a depiction of the DNA. And this forms an active complex so that EWS-FLI1 can go and do all of its transcriptional manipulations. TK216 is a drug that actually binds to EWS-FLI1 and blocks the interaction between RNA helicase A and EWS-FLI1 so that this complex can't be formed, and then EWS-FLI1 cannot affect the transcriptional activity of the cell. And so there's some really nice preclinical activity of an analog of TK216, which comes out of Dr. Turetsky's lab. The drug is called YK4279. And there have been a number of papers published on this, so I'm just going to kind of show you the highlights. But basically, when you use this drug and you measure EWS-FLI1 activity, you can see that with increasing doses of the drug, the activity of the fusion protein goes down. And what this translates into is a number of problems for the cell. So here I'm just showing you one readout because I already talked about it. So this is gamma H2AX again, so DNA damage. This is a control cell. This is a control group of cell. This is cells treated with the YK drug, and you can see that there's more of these sort of brown spots showing that there's more DNA damage in the cells treated with YK279. And we also see this increase in cell death with YK279 as well. In this particular experiment, they compared it to vincristine, which is a standard of care drug for Ewing sarcoma, and you can see that I'm just going to call it YK. YK has about as much, if not more, DNA damaging capacity as vincristine, and that when you combine the two drugs together, you get even more. So there's some sort of additive effect of the chemo plus the YK drug. And this also translates into superior survival in mice that are treated with this particular combination. So again, this is a survival curve. This is the proportion of mice surviving over time. And you can see that, the red line and the green line are showing you 2 doses of YK plus vincristine, which the mice survive longer than mice treated with vincristine or YK or nothing. So it's also very exciting to report that TK216 is also under evaluation in a clinical trial. And this is a phase 1 dose escalation study in patients just with relapsed and refractory Ewing sarcoma. And the sort of 2 phases are phase 1A, again looking what is the maximally tolerated dose, and then what is the maximally tolerated dose when added to vincristine. So this is a really novel study design where we're not just looking at one drug alone, but we're able to combine it with a chemo agent. And then phase 1B is then what is the response rate and duration for expansion cohorts in TK216 plus vincristine. And so far, 29, maybe 30 as of this week, patients have been dosed. This study is also still in dose escalation at dose level 9. And Dr. Turetsky tells me that this is equivalent to the efficacious dose that they saw in the preclinical rat model. So hopefully we're approaching where we should start to see some real activity. And the toxicities overall have been mild. This is unfortunately not open locally, but it's a multi-center study that's open throughout the United States. Okay, and the last example that I wanted to touch on is the anti-PD-L1 antibody atezolizumab for alveolar soft part sarcoma. So this is a drug that falls into our immunotherapy class. And I wanted to just step back and tell everyone what we mean by immunotherapy. Sort of the most general definition is getting the immune system to recognize cancer cells as foreign and destroy them. And there are a lot of different ways to do this. So we can manipulate the immune cells. So for those of you who've heard of CAR T cells, we take the cells out of the patient, do something to them, and put them back so that they can recognize the cancer cells. or you can activate the immune response in the patient. And there's multiple ways to do this. You can do it with viruses, adding cytokines, vaccines, or with checkpoint inhibitors, which is what atezolizumab is an example of. And so atezolizumab is an antibody that binds to and inhibits PD-L1 from binding to the T cell surface protein PD-1. And when you have PD-1 and PD-L1 binding, they suppress T cell activity. So this means that potentially T cells cannot recognize tumor, or even if they can, they don't do anything about it. And this is just a graphical description of this. So here's the tumor cells up here. This is the T cells down here. And so when you have PD-L1 binding to PD-1, it suppresses the T cells. If you put an antibody like atezolizumab that binds to PD-L1, it interrupts this binding to PD-1, and potentially that can activate the T cell, and that's what we call cancer immunotherapy. So this story is a little bit different than the other two that I described. So those really started at the bench, and I showed you the preclinical work that led up to the trial. This story begins with an observation at the bedside. So in an early safety study of atezolizumab in patients with solid tumors, one of those all-comers studies that I told you about, there was an adolescent patient with alveolar soft part sarcoma who was treated and had a marked response. So this was a patient who had complete resolution of a big lesion in the brain. And alveolar soft part sarcoma is known to be resistant to lots of standard chemotherapies, and there were no known curative options for patients with metastatic disease. So this was a really exciting finding. So subsequent work was done to look at the expression of PD-L1 on alveolar soft part sarcoma cells. And in fact, just to make sure that this one patient wasn't an outlier, in fact, what was found was that actually PD-L1 is highly expressed on alveolar soft part sarcoma. And I will say this is a very rare disease. There's not a lot of research being done on alveolar soft part sarcoma. And so, at least I wonder what would have happened if we didn't have that patient in that early trial. We may never have known that this was a disease that could be sensitive to this. So it sort of speaks to why clinical trials are so important. So based on this alone, there's now open a phase 2 study of anti-PD-L1 antibody atezolizumab in patients with alveolar soft part sarcoma. And it's a really interesting study because there's a phase 2 adult cohort, but there's also a phase 2 pediatric cohort for patients 6 to 18. And essentially just looking at the response rate in the adults and the patients. And so the study progress update that I'm going to give you is on a recent patient that our group has seen. So this is a 12-year-old girl. She enrolled in January. She came to us with a lot of lung disease. So these are all, for those of you who are not used to looking at these, this is a CAT scan of the patient. This is the neck. This is the belly. There are her arms up in the air, and these black areas are her lungs. And what we should see in the lungs is essentially all blackness. So everything in the lung that looks like gray blobs, this is all tumor. And this is a very classic appearance for alveolar soft part sarcoma, which typically metastasizes to the lungs and the brain. So she went on atezolizumab, and after 2 months, we scanned her again, and it looked like maybe we're starting to see that there was a little bit of improvement in some of these, but it wasn't particularly dramatic. We kept her on it, and as of this past month, she's shown marked decrease in the lung lesions. And so this patient remains on study, but we're really hopeful that we will get her to a complete response. And so this is currently recruiting patients. It's a multi-center study for the adults, but for the pediatric patients, the only open site is at NCI. So if anyone has an alveolar soft part sarcoma patient that is looking for a therapy, we would welcome your patients. So I just wanted to end by giving you this slide again and reiterating that I think that there's much to be hopeful about, despite that depressing FDA slide I put up earlier. All of these things are open throughout the country. And one thing that I think is actually really exciting is that everything that I've highlighted here in yellow is open locally at many of our institutions. So there's a lot out there for patients, hopefully a lot to be learned through these clinical trials, and hopefully a lot of progress to be made. And so I wanted to say thank you first and foremost to the patients and their families and advocates, because obviously we couldn't do any of this work without you. The members of my lab, in particular Anna Gibson and Josh Baumgart, who did the work on Pen 866, members of the POB, the UOB, the DTB, and the DTC at NCI, and of my former mentor, Lee Hellman at CHLA. And then, of course, thank you to the Children's Cancer Foundation for supporting my work and the work of so many other young investigators. And I'd be happy to take any questions.
So we think back to like 10 or 15 years ago when we were talking about HSP90 inhibitors in the laboratory and thinking about how, as the chaperone protein that HSP90 is, it sort of depends on the degree of sensitivity of that client protein to HSP90 inhibition, right? And so some of the earlier data out of Rosen's lab and some other labs showed that, for instance, mutant EGFR had a higher dependency on HSP90 than wild-type EGFR, and the same was true for mutant BRAF, for example, right? And so I always thought of HSP90 inhibitors as having the greatest effect in tumors where you could demonstrate that the mutant oncoprotein was a selective HSP90 client. So when we think about using it in sarcomas, one, do we have any idea what the true HSP90 client proteins in the cell are? And two, do you think it's working at all in your experiments by true HSP90 inhibition, or is that just simply a way to keep the irinotecan in the cell longer and allow it to have a more profound effect?
Yeah, so I'll answer your first question first. So I think we have some idea of what the client proteins are. We actually have done some work in our lab on HSP90 inhibitors, and that's how we got into the conjugate initially. So for example, like in rhabdo, we see, loss of IGF-1 receptor expression, things like that. However, I think in this case, the HSP90 inhibitor is not acting as an HSP90 inhibitor because it's very hard to look at this particular drug in vitro because it doesn't have the same activity, right? But we have access to the HSP90 inhibitor that is used in the conjugate. It's called 8663. And when we use that in vitro, we actually don't see a downregulation of any of the client proteins, and we don't see a compensatory induction of HSP70 either, which is a marker of true HSP90 inhibition. So I think in this, and that was, I think when the company was developing this, they actually had a number of different conjugates with varying potency for the HSP90 inhibitors. And they saw similar activity of the sort of delivery method as opposed to across the HSP90 inhibitor. So it didn't really matter the potency of the HSP90 inhibitor to induce HSP70 or to downregulate any of those client proteins. The drug delivery effect was the same across the board. So the clinical candidate drug that went into the clinic actually is a really weak HSP90 inhibitor. I think in part because they didn't think they needed a strong one, and maybe that just buys you toxicity and no improved efficacy. So my sense is from our work and from the work of others that it's really just acting as a delivery, like a kinetic trafficker for the overall molecule.